Alright, today guys, we're just going to be going over some of the young earth creationist lies, specifically about mutations. So this is going to be a series, but we're only going to basically just cover mutations today and a little bit of basic biology. So, biological evolution, what do we mean by this? Well, for creationists, let's start off by talking about what we don't mean by that, right? So, what we don't mean is how life came to begin. What we don't mean is how the universe came to begin. What we don't mean is how inorganic chemicals change. What we don't mean is how the planets and celestial bodies came to be how they are now. What we mean by biological evolution is how populations of living things change from generation to generation via inherited modifications, which produces populations with diverging sets of traits. Now, time is going to be a big issue here because over millions of years you would expect these diverging sets of traits to be far diverged. Creationists claim that these traits are not far diverged because 1, they're not related, and 2, the Earth is too young for that to happen. To justify this belief, they cherry-pick quotes and sciences to selectively support their literalist interpretation of the book of Genesis. They'll learn a bunch of terminology and jargon and try to dazzle you with complex explanations, but they don't actually know what they're talking about. This trick only works on laymen. But let's honestly engage with them, because not all of the creationists are scammers. Some of them are being scammed. Probably most of them. So, how are you supposed to get a fish to evolve into a fisherman? Now, this isn't actually a straw man at all. But it's a strange question because no one disputes that there are fishermen among the fish. Sharks eat other fish. Some fish don't eat other fish. How did we get this variation? Are we already saying that God has separately made sharks from fish? This is not typically how we think of evolution. But this is how creationists imagine it. They don't understand the gradual differences or the gradual change. It's a gradient. It's not popping from one to the other. You're not getting one creature coming out of the other, or a totally different type of creature coming out of the other. Parent and child are always going to be the same species. So, if you can see this diagram here, with the gradual change of color, there's no point at which you can say it hopped from red to green, because it never did. This is red, it's still kind of red, it's orange, and it's red and yellow. And now it's just yellow, and now it's becoming green. So, this is what we're talking about here. Creationists accept that all these cats and all these birds have been speciated via genetics and breeding. And they accept the comparative anatomy and the comparative genomics between these creatures, because it's pretty obvious that they share 99% of their characteristics. Yet when it comes to apes, the creationists have some kind of a double standard here. And so when we're talking about speciation. They accept speciation. They accept microevolution. Nobody disputes that that kind of diversification happens among living organisms. Creationists accept that this happens. Half the battle is already won. Scientists just reason that this change can be traced back further using the fossil record, in which we see links between different kinds, which fit neither category. And again, we see something more like this gradient, this branching gradient, than anything like this. And this is not what we'd expect under the special creation model, but we'd expect something just as ridiculous, which is that both of these creatures are just being created independently. Now, the problem here with the creationist model is that they can't distinguish between a created kind or a created trait and an adaptive trait, or something that has micro-evolved into the population. So, even though these cats all share most of their same traits, some of the cats have developed traits that none of the other cats have. And this happens all throughout life systems. We'll go over a few of them, especially in part 2. So they agree with microevolution, speciation of the existing clades, but they reject macroevolution, a gradient between clades. We should note here that the genetic analysis, the comparative genomics, creates almost the exact same tree as those created by comparative anatomy. We usually use the genomics to correct the anatomy trees because genomics is actually tracking an inheritance of a gene, whereas with phenotypic comparisons, we're just seeing the result of a gene. So it's a less direct method. But they both confirm each other. So again, just going to drive home, this is going to be my main point. For the majority of these debates, not for today's discussion, but I'll drive it home anyway. Look at all this diversification between bird kinds. Is this what the Bible meant? We're not going to get any specifics, and these three are considered not true birds. This is. But you would never say that it's not the same kind of creature as these. In fact, the chicken is more similar to the velociraptor than the penguin is to an eagle, or an owl, or a kiwi, or a peacock, or a swan. And it's clear that these dinosaur feet and these features that were developed on dinosaurs, the claws, the scales, or not the scales, the feathers, they accept this kind of diversity, but not this. So they have to double down, and they have to actually put themselves in a worse corner, in my opinion. And what they have to do now is either decide that these are all related, or decide that all these birds, they're not related. Some of these birds are specially created, but the problem here that they're backing themselves into is, again, they're asserting this, but they have no way of telling you which is which. That's a fundamental contradiction that they can't get past. They're going to assert that these are different kinds, but they're not going to assert how they know. And that's how we know that they're full of crap. This is the classic God of the Gaps arguments. They fill any gap in scientific knowledge with their God. And the problem with this is that it's an ever-shrinking gap. Your God, you are putting your God in a place that is ever shrinking, and you're making your God ever more pathetic. And it's not even good science. Just take this picture as an illustration. You're doing math, and to bridge these two gaps, or to bridge these two things that you know, that are quite specific, you just say, some magic happens, a miracle occurs, and we get this. That's not specific enough to be science. So creationists want the legitimacy of the word science, but they want that legitimacy without doing the work to pass the classes, without playing by the rules of naturalism, without actually doing science. They just want the word. They just want the label. They don't want to do the action, because science gives results and they do not. Otherwise, they could rely on their own reputation instead of borrowing the terminology of a contrasting worldview. There's a reason they object to characterizing their beliefs as just a belief in magic, because they know magic as a concept has no credibility and no explanatory power. They want the gravitas and the respect that comes with being a serious academic, while telling all the much more highly educated experts that they have it all wrong. Creationist science is stolen valor. They're also quote miners. Stephen Jay Gould, I've heard cited many times by the creationists. But they never finish his sentence. The fossil record with its abrupt transitions offers no support for gradual change. ... And the principle of natural selection does not require it. Does not require gradual change. Selection can operate rapidly. Now, this may seem to contradict with what I'm saying over here about this gradual change. That's just a difference in word choice, essentially. What he's talking about is that this is actually an oversimplification. We don't actually see gradients in animals like this. This is to illustrate the kind of animal. So, for example, what Stephen Jay Gould is talking about is you could see an animal, a cow-like animal with little nubs on its head. Its child might have big horns, the first horns, where its parent did not. It just had little nubs. And that seems like a big jump. You don't need to have every single transition, every single centimeter accounted for as a transition. But what this is illustrating here is that it's still a cow-like creature, or it's whatever, it's the precursor to a cow. They're still the same kind of creature. Even a big difference in the horn size is not going to change it from whatever it is. So that's just like adding a little bit of yellow into the red. Do not conflate gradual change of a specific trait with the gradual change of a population. Okay? That's a mistake. And if any creationist is confused about that dichotomy, if I haven't done it justice here, please take it up with me. I would love to discuss it in detail. So, it may appear as if we're arguing over the same evidence. I mean, we are, but both arguments are not equally valid, because really we're not arguing from the same evidence in the end, since the creationist misrepresents the scientific conclusions about said evidence. The young Earth creationists are essentially making all of the exact same arguments as flat Earth. God made it. Science is biased against supernaturalism. Science is an atheist conspiracy, or at least, the scientific consensus. Science as we know it is a religion known as scientism. can only be supported by biblical passages and is obsessed with biblical passages, and is widely considered to have no validity or predictive power by 99% of the experts. And this applies to both young Earth creationism and flat Earth. They're making the same arguments, especially when it comes to not trusting the scientific consensus. I think I tend to agree with flat earthers when they say that the Bible supports a flat earth model. So if young earth creationists are going to make all this stink about how you might be in trouble on Judgment Day for not believing in creationism, even though supposedly faith in Jesus is all you need for salvation, why aren't the flat earthers, or why aren't the creationists so concerned about following the flat earth model? It appears that even creationists have at least that much sense, but the lack of consistency is certainly disappointing. Now, life could have been created. Created life still means evolution. You can look at it as an evolving software. Most life scientists do. Most life scientists are Christians. I don't think most physicists are. But they believe that life was created, but then it evolved. This program, whether natural or otherwise, this DNA, evolves on its own. This much is undeniable even by the staunchest creationists, because you agree with speciation. Adaptation, diversification, natural selection, speciation, this is all evolution. Microevolution, you'll say, But, so the true point of contention would be mutation and, of course, the accumulation of these microchanges that occur from microevolution. Creationists clearly agree with speciation and microevolution. They claim for no reason that the change stops there. They also love to claim that such complex things cannot change. But we have programs which show greater propensity to improve themselves the simpler the programming is. Genetic code is simple. Four bases combine to code for amino acids which can create almost any molecule that life needs. We also need fuel, of course, like oxygen and other things, but we actually get most of those nutrients from other life. Just to go over the program analogy again, just to show scholarly evidence of using evolution to push the boundaries of programming. And you'll say, but you're using intelligent design. And no, that's exactly the point. They're actually making their programs self-improving by taking out some of the designed information and making it simpler so that it can actually teach itself and learn itself. So again, evolution is totally compatible with life being created by God. But the science is not compatible with the supposed Noah's Ark kind of animals being created by God. It's quite clear that the first life, no matter how it got here, was single-celled. This is just the actual citations from the guys, several people who have actually created self-improving programs based on evolutionary principle. So how do we get a cow from a non-cow? Again, this is a straw man, but I will attempt to address the real question behind this misunderstanding of evolution. Even if you could point to the first true cow, its parent would be virtually indistinguishable from a cow. If the cow population were to evolve into something else, it would be very close to a cow. The population would have to undergo millions of years of these changes to no longer look like a cow, but it will still contain the cow lineage and will still contain features that the cow had. So, Do not strawman us, please. I don't know. The creationists won't stop on this line of how do we get a cow from a non-cow, how do we get a dog from a non-dog. But all we can do is just point out that that's not evolution over and over and over. Hopefully one day they'll stop. I know they won't. And that's why I'm preempting this, because I know they're going to say something like that. So once you've gotten past that with them, and you occasionally can, it won't last, but you can do it. You can get them to agree temporarily. It's just a cascade of moving back the goalposts, because that's what a pseudoscientist has to do. They can't ever put their foot down on something, because otherwise that would be a fact and they could prove it. They tacitly admit evolution occurs, whether it's evolution of the original biblical kinds into the diversity we see today, or whether it's evolution towards dysfunction and death by mutation accumulation. They admit that there are mechanisms that change population of organisms through the generation, and that is evolution of life. Period. End of story. We're going to contend on what the first life was, and so that's what I'm going to try and just demonstrate to you with this series and with these debates. But this is going to start specifically with debunking this theory of destruction of all life by deleterious mutation accumulation on the onset of the fall of Adam and Eve. So, starting with the first goalpost is the most generic. Evolution is false. You've never seen a step towards a new kind of organism. This kind of assertion is false because we have several experiments, most notably in the algae species Chlamydomonas reinhardtii. This species is a single-celled algae, and as you can see, it will replicate and then separate and live as single cells. We have done several experiments that have subjected these algae to selection pressures. In this experiment, they were selected to sorting pressures according to size by being put through a centrifuge. So the single cells got crushed, and the colony cells were able to stay more on top and not get crushed by material that was on top of them. In the second experiment, this is more straightforward. The same exact species was subjected to predation selections by a specific predator, another single-cellular organism, the paramecium. So the paramecium could eat single-cellular algae, but it could not eat the colonies. So the colony populations took over. And these are multicellular organisms because they operate as colonies. Now, I'm sure you'll ask, so how do organs develop and how do cell specialization develop? It's quite simple. We know that even the cells of complex organisms all have the same DNA. And their DNA is being regulated by the cell itself sensing the outside, and by the cell communicating with other cells, whether it's immediately or through systemic hormonal changes. These cells regulate each other's functions. So, since the communications between them can differ, their functions can differ. And we've observed this. We know this can happen. So it's not a stretch to say, okay, well, next step, after we've evolved a multicellular organism from a single cellular organism, and we've observed this in real time several times from wild existing organisms, well, it's not a stretch to say that these cells can now communicate with each other and thus regulate each other's behavior. But we're focusing on mutation for now. So the creationists will instantly recognize, well, if we're going to have evolution, well, how does that proceed on the basis of random mutations? Creationists will claim mutation cannot drive evolution. An honest person would ask questions, how does it happen? Creationists simply claim that it can't happen, even though they've been shown to be wrong. Most evolution is driven by breeding and mixing of already existent traits, but alleles, which are different varieties of the same type of gene, are arrived at through mutation. Some mutations will cause loss of function, and that organism will usually die or be left out of sexual selection. But there are some mutations which just simply cause a change of function. A simple example would be a silent duplication, where you would get one gene duplicated. So these can be bad, but typically the worst that can happen is you just have an over-regulation or something. So a simple example, you might just be really hairy because you might just be producing extra hair gene, right? Extra keratin protein. But the advantage of this is now you can evolve a new gene from that extra one that you had. That can now mutate. And there's this concept called a hotspot, which is genes that mutate tend to mutate more than the other genes that don't mutate. So if you're getting a duplication in an area, it's more likely that gene will mutate again. So it's not exactly random. And the reason for that is it's occurring at a place where the DNA is unstable, and that's associated with a specific code, so those occur in the gene. We've seen this happen with antifreeze protein genes, which help fish and insects not freeze their blood. Then creationists will deny that mutations can cause incremental changes to the body plan of an organism, such that it moves towards a new type. We've already discovered the Hox genes that are responsible for the shape of the spinal cord and invertebrates and limb development in invertebrates, like insects. So this is a gene that's coding for the structure, literally the core bones, and an insect's bones is its exoskeleton. So we know these genes are actually doing the same thing. An insect wears its bones on the outside, we wear our bones on the inside. But we see the same type of gene in insects and in humans. They're just slightly different. And chemical and physical differences in proteins are what create physical differences in body plan, not just a difference in blood serum. See, a body part will continue to grow if its cells are getting signaled to grow. It's not like a blueprint. It's not like they're reading, go 3 centimeters this way and then stop. That's not how it works. It's all just, you keep getting the signal and you stop when you stop getting the signal. And the signal is regulated by the gene. So the signal will turn off at an arbitrary time. Genes that turn off at the right time will be conserved in the population. Body plan is also changeable simply through breeding different traits that already exist. This is the skeleton of a penguin. Its wings and its feet have a new function. Its whole body has a new function. Its feathers have a new function. Same body plan. Same bones. In embryos, we see these structures that are similar to gill slits in fish embryos, and all embryos have this. But they turn into different things in different organisms. They turn into gill slits in fish. Incremental changes to body plans occur among kinds. What kind of change would qualify as a difference in kind? Seriously, this is one of the very important questions. If a creationist is watching this and wants to pipe in, this would be a great time. What kind of change would qualify as a difference in kind of either frog or amphibian or any kind of animal? What is the minimum change that would qualify for you? Because there are drastic changes within the exact same species. But we don't need to qualify that. Incremental changes to body plan occur in the same species, just turning on and off different genes. Why is it a stretch to say the same thing can occur through breeding and evolution? The fourth goalpost, creationists will say there is no way to build a genome, i.e. add new information. We've gone over this, though. Duplication is that way. Well, they'll say, well, that's not new information. You're just adding more of the same stuff. Well, if you want to go at it that way, then the genome has never added any new information because we're all just working with the same 4 base codes. That's it. These same 4 base codes can be gained, lost in different combinations. It's these recombinations. Loss or gain of information can recontextualize old information, just shuffling it up. If you put a bunch of Scrabble pieces in a pile and shuffle it up, eventually you're going to see a word appear in that. It's kind of how word searches work. Remember, also, I'm sure most of you have played with Legos, and I'm sure most of you, your original sets didn't tend to stay together. They tended to all go into the same pile, and then you would make new creations from all those pre-existing bricks. You didn't have a little 3D printer making new bricks for yourself. No, you were just making recombinations. New information from the old. This is just going over kind of what I was explaining. That you can duplicate and then mess with the duplicate. If you just keep accumulating nonsense letters, eventually you're going to get something that makes sense. You're going to get combinations that are going to be able to, or you're going to get enough variance that you're going to be able to make combinations that make sense. There's this classic thought experiment where you can never get a meaningful text like Shakespeare's Hamlet from just monkeys randomly banging on the keyboard. And it's usually posited as infinite monkeys, infinite time. Eventually one monkey will type out the whole thing without any mistakes. Yeah, I mean, that's a different thought experiment. If you wanted to make an analogy for evolution, you would only let the monkeys type one letter at a time, and then you would proliferate each step that was correct. Every time they put the right letter down, you would stop the experiment, replicate that page, and give it to all the monkeys. And then one of them is going to type the next letter. They have a 1 in 26 chance or something to type the correct letter next. So you can get all of Hamlet with no errors in like 637 days. That was my calculation. And this was done realistically. 5 minutes for each typed letter, copying the paper, and then putting it back in each typewriter. So, once you've dazzled them with your science, they will step back and they will say, well, sure, but mutations cannot lead to novel functions. Mutations can only change or unlock the original function. But that's just not true at all. The antifreeze protein we were talking about earlier is derived from a digestive protein. Or a gene that codes for a digestive protein. So you fold the protein a little bit differently and it has a different function. Might still have all the same amino acids or most of the same amino acids, most of the same functional groups. But maybe new functional groups are exposed. Maybe the protein has a new flexibility. The reason we know that it's derived from a different protein is because there are fish. All these fish have the digestive protein, and then there are certain fish that have this antifreeze protein. And what do you know? Its gene has information that is excised from the digestive function protein. So we're seeing different proteins derived from the same gene by recombining the information. And these proteins have different functions. So this goalpost gets knocked down as well. But let's keep going. Whales evolved a new function for their teeth. Do you think that baleen has the same function as any other tooth in this world? No, it doesn't chew, it doesn't bite. You're going to say, oh, it aids in eating. That's not the same function. That would be like saying wings and feet have the same function because they both get you somewhere. No, baleen does not have the same function as teeth. But it is made from the same stuff and the same gene as teeth, and we've proven this. A different fold or flex site could change the function of a protein, destroy its function, or retain old functionality plus a new ability. And this goes towards this concept called anti-fragility, which we will talk about going forward. So since they've moved the goalposts back again, they'll now say, well, you can never gain a new function from loss of information. And that's simply not true. You can lose limbs and still have a different function to your body. For example, whales and snakes. Their body has a different function. They have lost limbs, though. Humans also have lost... Yeah, we'll get there. You know, how is loss of hair and limbs not a gain of a hydrodynamic function to the body plan? How is this guy not gaining a new function from his worm-like body versus crawling with limbs? You know, and young Earth creationists will talk a lot about how large genomes are unhealthy, or that, you know, we're just losing information. Well, that's fine. None of that contradicts evolution at all. You know, your absolutes are not true, but even if they were, they don't contradict evolution. The amoeba has the highest genetic load of any known organism, and they survive just fine, and it would seem, well, they are remarkably simple, but they have a lot of genes. it would seem that a lot of information might just kind of make you a blob, and that loss of information can kind of carve out specific traits and specific adaptations, much like and shaping you through pressures, much like shaping a statue out of an amorphous rock. Even assuming the genome cannot grow or create new information, it is easy to see how loss of genetic material can carve out specific traits from a formless precursor. So even if we're assuming that you cannot gain new functions or information, even if we're assuming that all genetic information is encoded in the first life form, our fossil record still makes sense. I mean, some of the genetics might not make sense because you're biochemically wrong, but that's how robust the theory of evolution is. Anyway, creationists will then say beneficial mutations cannot occur from or respond to loss of information. So I'm going to repeat again, snakes and whales lose their limbs to their benefit, apparently. I mean, it's not like the limb was necessarily a burden for some type of environment, but clearly the snake can go in an environment that a lizard can't. Anyway, this is really more about deletions causing beneficial mutations. We actually see that between humans and apes, there are 510 deletions, which are associated with brain growth. Quote, another deletion removes a forebrain subventricular zone enhancer near the tumor suppressor gene growth arrest and DNA damage inducible gamma. Oh, that's just another study. Removes a forebrain subventricular zone enhancer near the tumor suppressor gene. A loss correlated with expansion of specific brain regions in humans. That's from human-specific loss of regulatory DNA and the evolution of human-specific traits. Here's another study. The point is in the title. Loss of function of Saccharomyces cerevisiae, kinesin-related SYN8 and KIP1 is suppressed by CAR3 motor domain mutations. So, what was that? What did that mean? It means that we had a protein, a kinesin-related protein, lose function, and that loss of function is suppressed by a different mutation than the mutation that caused the loss of function. And when loss of function is suppressed, what that means is it gains the function back. So we can also see chromosomal fusions without necessarily having disease. And that would be loss of information as well. Antibody production, if you don't know what antibodies are, those are the little things that tag bacteria and viruses and fungi and parasites inside of you, and they attach to those and they mark them for destruction for the immune cells. So antibody production necessarily involves induced mutation by loss of information and recombination. So the variable regions of this gene, or of this antibody protein, consist of several possibilities, taking one region from 3 classes of the gene and recombining them, discarding the rest of the gene. Coding genes lose information called introns. That's what these are, essentially. So, there are these regions. The gene selects one from each region randomly, puts them together in a new combination, because these are all just randomly selected, and it gets rid of the rest of the information. So, now, this is just an example of something that I have personal experience with. I actually did this. Loss of information leads to a new function. a programming example. So the top example, I didn't write the script, but essentially what it codes for, this isn't the whole thing, but this is the functional part that we're looking at. What this did was, it was a capture the flag kind of deal, and it was a countdown to the win time. So, and the countdown would pause every time the defender team would recapture the flag. So I don't actually remember what I was doing. I think I was just randomly, I think I was trying to take things out to see how it worked. And I took this bit out to see if it would do anything. This was me teaching myself how to code. This is how science is done, by the way. You perform experiments and you see what happens. So what happened was the flag functioned just the same, except the countdown did not pause. It reset back to the total every time the defending team recaptured the flag. So this was an entirely new function, and I used both of these flags for different types of games, different types of gameplay, different functions, from a deletion of information. So, even though we've shown a long list, maybe this goalpost should be earlier, but creationists will claim mutations can only lead to dysfunction. This is simply not true. Not in all cases. They use the fact that it can lead to dysfunction to say that it will lead to dysfunction. Translocations can lead to dysfunction, for example, mixing up of whole chromosomes. They can be unbalanced, or they can be balanced. This simply refers to whether or not the cell will be able to divide the chromosome evenly during cellular division, so that it will be able to divide again without problems. Such translocations are usually harmless, and so the ones that aren't sneak by. The ones that are either don't get born or have such problems that they either can't reproduce, so can't pass on their bad genes, or they won't get selected for by a sexual mate. We'll go over in detail sometime human and chimp relations, or not human and chimp, but human and ape relations. And hotspots. Hotspots seem to be one of those pseudoscientific traps for creationists. They'll claim things like there are 0 recombination hotspots that are common between apes and humans, which, we wouldn't necessarily expect there to be, because hotspots move, because that's what a hotspot is. It's gene recombination, and recombination can involve the movement of genes, so we expect hotspots to move. But creationists will do things like claim, without any sources at all, that there are no similarities in the hotspots. There are. But we observe differences in hotspots between species as well, such as dogs. I actually will, I'll put that citation in the description, because I don't have it here. So really just to touch on some of the more dishonest things creationists will try to spin. They'll lie about hotspots, and then they'll lie about the different, the gap. They lie about how big the gap is between humans and apes. So the average nucleotide difference is 1 to 2%. That means not even the amino acids are necessarily different, just the nucleotides, because you can get different amino acids from, or you can get the same amino acid from different codons. Just for example, alanine can be coded for by GCU, GCC, GCA, GCG. Those are the ones, yeah. 4 different pairs of 3 nucleotides can code for the same amino acid. So if the difference between nucleotides is only 1 to 2%, we're probably going to have mostly the same amino acids. And so this is what we're looking at over here. Creationists, I've seen cite the study called 80% of proteins are different between humans and chimpanzees. And if you only read the title, you might come to a creationist conclusion. But in the study, it goes over how the percentage difference is never more than 2%. So 22% of enzymes are 100%. Well, you can read this, right? Percentages of proteins showing 100%, 99%, and 98% sequence identity. We're seeing most proteins have 100%, 99, and 98% similarity. And that's on amino acid level. That matches up to the nucleotide level. And a minority of proteins have less than 98% similarity. So most proteins have 98 or higher percent similarity between apes and humans. This study is saying any nucleotide difference is a difference. So 20% of proteins between apes and humans are exactly the same. No nucleotide difference whatsoever. So, at this point, the creationists will start saying things like, if evolution cannot fully explain every single microchange in every organelle, it is false. For example, how sex evolved. So we don't have to know how every organ evolved. We really just have to know about 1 or 2 evolutions, and we can see that since the populations are changing progressively through the rock layers, and we know that organisms and populations change, we know that they're changing over time, that they're evolving over time. So we don't have to know how one specific trait evolved, as long as we know that traits are evolving. And again, we don't expect to see a perfect gradient of every trait. We're just seeing a gradient of population. And again, this is an attempt to circle back to the God of the Gaps argument. Because if we can explain 90% of the trait change, but we can't explain how this other 10% of traits changed, then they're going to claim God of the gaps. They're going to say, well, you can't explain that, therefore God did it. That's not an argument. That's the most intellectually lazy argument I could think of. It is a fallacy. It is saying, you don't understand how it happened, therefore it didn't happen. And once they're at this point, creationists are going to get really desperate and start to leap back to the beginning again, trying to put a biogenesis on us, saying mutations cannot give us the fundamental DNA code and transcription proteins. This is fair. This is a fair misunderstanding. If you don't understand mutation or evolution very much, you're going to think, well, I guess the evolutionists are saying that the very concept of DNA itself mutated and evolved. Perhaps there's some chemistry to suggest that, but that's not a claim made by evolution. Not at all. This is that's assumed by evolution. Whatever is necessary for life is assumed by evolution. Evolution only studies the change of living organisms. It does not study how living organisms began. So, please do not make this conflation. No one claims evolutionary processes originate these structures, except creationists. So it's a straw man. And again, evolution leaves plenty of room for a divinely created cellular machinery. But it should not be assumed that God must be the only possible cause to an unknown. That's special pleading, and it's an argument from ignorance. So, the final goalpost of what we were trying to talk about this whole time, the red herring. This actually doesn't really matter to proving evolution. I just have a stick up my ass about it because I'm a biologist and I know the science, and the creationists are able to trick people into doubting evolution based on bunk, fake biology. So I'm going to take away that ammunition from them here. It's also used by them as a distraction so that they don't have to talk about transitional forms or comparative anatomy. They just can try and poke holes in the science of mutations, which they can do successfully because it's a very complex science. Not many people are very well trained and are able to spot the lies. But so let's go over the legitimate science. Accumulation of deleterious mutations is evolution. Even if we're going to assume the creationist model of absolute accumulation of deleterious mutations, this is still evolution. Again, this is how robust the theory is. We're only looking at change of living organisms. Okay? So even if the change is towards degeneration, towards dysfunction and disease, well, guess what? That's where evolution is heading towards. That's where the change is going. So to get to this concept of genetic entropy that they use, it's not actually a scientific term, but I'm speaking to creationists, so we'll speak in their terms. This basic concept essentially exists. However, what young Earth creationists refuse to accept is that there are forces which work against entropy and organize things in the other direction toward order without any intelligent guide, and this is the base of a lot of sciences that deal with equilibrium. This is called disequilibrium, and it can go in either direction. It can go towards entropy or towards order. And most people object to conflating genetic entropy with physical or thermodynamic entropy. And that's true, it is a mistake to conflate those, but of course I understand it as a metaphor, but the metaphor holds because, again, there are forces that behave like entropy and behave like order when we're talking about genetics. So there are forces which can resist the forces of entropy. But every time we list these out, they go in one ear and out the other. Genes can fall into dysfunction or loss of function if there's no positive selection for them, like loss of functional eyes or pigments from creatures living in the dark. But again, the creationist factors only degenerative forces into their equations, ignoring willfully the creative processes of positive and negative selection pressures. Evolution started off with Darwin, obviously. The origin of species. He was looking at speciation. He was essentially noticing microevolution. And then he extrapolated to macroevolution. Darwinism has been abandoned. But that does not mean it was strictly wrong. Darwinism was incomplete and insufficient because it didn't have genetics. But Darwin's theory was confirmed in just 2 short years when his predictions that we would have a transitional fossil between dinosaurs and birds that would have unfused finger bones and teeth with a bird-like skeleton. 2 years later, we found the transitional fossil, Archaeopteryx. Isn't that neat? We can make a specific prediction and then go find specifically that thing. We don't have to retrofit and make the story fit what we want it to be. So, as our understanding of change over time changed over time, the neutral theory of molecular evolution developed. We started noticing that advantageous mutations rapidly fixed in the population, but are rare. Disadvantageous mutations are not so rare, but rapidly eliminated from the population. And then there were these other types of mutations that accumulated because they weren't under selection pressure, and we called these neutral mutations. And then we went to the model of slightly neutral mutations because we were seeing that they did still have an effect. They weren't just silent. They just were not necessarily visible to selection pressures. And over the years, we've discovered that only about 10% or fewer of mutations seem to behave as slightly deleterious mutations. So only about 10% of the time is something going to go unnoticed by selection. Now, the creationists would say, well, then you're going to have an unlimited buildup of nearly neutral mutations. There's no way for selection to see them. Well, there are models, there are methods, such as compensatory mutations, which basically turn them beneficial. The creationist problem is not entirely unfounded. Where they make their mistake is in their absoluteness. They're pointing to a specific phenomenon that happens that we call Muller's ratchet. This mechanism is how we get slightly deleterious alleles that become fixed in a population. And this is the problem that they're saying that we have with evolution, is that we'd all be dead soon because slightly deleterious alleles will become fixed in the population, and that this is the curse from the fall in the garden. Young Earth creationists use this possible event to claim that it is the absolute fate of all organisms, but they have no means of demonstrating this pattern. They'll point to things like extinct organisms and genetic diseases, none of which they can say are accelerating. They have to ignore the effects and additive effects of counterbalancing mechanisms. They take Muller's ratchet in isolation and ignore everything else. They ignore all the other selection pressures. The number of mutations that actually becomes fixed is so low that it can easily equal or outweigh the mutation accumulation rate. The compensatory mutation rate. This is actually another scientific term, the genomic decay paradox. Primitive models of Muller's ratchet predict extinction will occur much earlier than all educated estimates of the ages of particular species, including young Earth timelines. We would have all gone extinct much before 6,000 years. We wouldn't have made it 6,000 years, okay? The genomic decay paradox is a paradox, but a paradox is not a problem necessarily. It appears paradoxical because if one stipulation is true, the other cannot be. Thus, either our assumptions about mutation accumulation are wrong, or our estimates on the species age are wrong. Seeing as how even creationists estimate species to be older than these primitive models would predict that they're using, their position is incoherent, and we used multiple overlapping dating methods to confirm. So we're assuming that our assumptions about mutation accumulation are wrong, so we scientists updated our model. And we began studying it. Muller's ratchet under epistatic selection. So we're trying to understand how we're selecting out or compensating for the buildup of nearly neutral mutations or slightly deleterious mutations. Quote, if deleterious alleles have synergistic fitness effects, then as the ratchet advances, the frequency of the best available genotype will necessarily increase, making its loss less and less probable. As a result, sufficiently strong synergistic epistasis can effectively halt the action of Muller's ratchet. Instead of being driven extinct, a finite asexual population could then survive practically indefinitely. So this is a guy that creationists like to cite a lot, Kondrashov, because he studies problems with mutation accumulation. But even he is saying that he's found ways for even asexual populations who don't have the variation that sexual reproduction gives us can halt the action of Muller's ratchet, which is the accumulation of slightly deleterious mutations and the fixation of them in the population. The creationists simply neglect this part of the literature, but they cherry-pick other parts of the literature to make it look like a bigger problem than it is. Quantifying the genomic decay paradox due to Muller's ratchet in human mitochondrial DNA. The resulting genomic decay paradox complements a similar threat from extinction due to mutation accumulation in nuclear DNA and suggests evaluation of unconventional explanations for long-term persistence. A substantial list of potential solutions is given, including compensatory back mutations, mutation rate heterogeneity, and occasional recombination of mtDNA. Future work will have to explore which of these actually solves the paradox. Nonetheless, the results presented here provide yet another reason to minimize anthropogenic increase of mutation rates. So basically what that means, this is just an example in the literature of several different proposed mechanisms that can halt Muller's ratchet in different ratios. As we'll see, the math will play out. It's also saying that this is a delicate balance. So if we start putting mutagens into the atmosphere, then we might see an extinction due to mutation accumulation. This threat is much overplayed by the creationists, though, because even asexual populations have found ways around this, such as in quantifying the threat of extinction from Muller's ratchet in the diploid Amazon molly. The Amazon molly is a small unisexual fish that has been suspected of being threatened by extinction from the stochastic accumulation of slightly deleterious mutations that is caused by Muller's ratchet in non-recombining populations. However, no detailed quantification of the extent of this threat is available. So, the creationists don't know how much of a threat this is. The scientists are studying it, and we find... Not only do we find fossil evidence that indicates quite clearly that we've been around for a lot longer than Muller's ratchet would, than the outdated model of Muller's ratchet would predict. These types of fish are actually all female, and they steal sperm from a different species of fish. So several biological processes could individually or in combination solve this genomic decay paradox, including paternal leakage of undamaged DNA from sexual sister species, compensatory mutations, and many others. Now, we'll talk about compensatory mutations. There's a reason it keeps coming up, because it's a powerful force. This one creationists love. Contamination of the genome by very slightly deleterious mutations. Why have we not died 100 times over? And they laugh at that. But the point of that question is, even by a creationist timeline, we should have died 100 times over. This is the next slide, so we'll get there. The fact is that we see variable rates of and locations of mutations. If accumulation were a problem, we should have died 100 times over, even in 6,000 years. This paper is just describing the dangerous range of selection coefficients, which can lead to accumulation of slightly deleterious mutations. And this is what we see when we look at when selection acts on different mutations independently, but this implies too high a mutation load. This paradox cannot be resolved by invoking beneficial mutations alone. So we understand other things. So it's not just beneficial mutations, it's not just compensatory mutations. Let's see what else is going on here. Several possible resolutions are considered, including soft selection and synergistic epistasis. So we will explore that. If creationism were true, we wouldn't see genetic stasis. We would see a continuous degradation with no living fossils. But we see constraints to the buildup of mutations to the point of stasis. This paper discusses the conditions in which stasis can break and go towards the direction of SDM accumulation. Young Earth creationists won't look at the math, so they won't realize that accumulation only happens in a dangerous range, which means there is a safe range. mutation accumulation and the extinction of small populations has reported that application of empirical estimates of the properties of spontaneous deleterious mutations leads to the conclusion that populations with effective sizes smaller than 100 and actual sizes smaller than 1,000 are highly vulnerable to extinction via mutational meltdown on timescales of approximately 100 generations. So yes, what's 100 generations on the human breeding patterns? Let's say every generation is 30 years, just to stretch it out real far. Let's see, 30. Well, I mean, this is simple math. 30 times 100 is 3,000. So we should have been dead 3,000 years ago, according to creationists. The metapopulation extinction caused by mutation accumulation. Theory suggests that the risk of extinction by mutation accumulation can be comparable to that by environmental stochasticity for an isolated population smaller than a few thousand individuals. So, unstable environments, small populations. This will cause Muller's ratchet. Here we show that metapopulation structure, habitat loss, or fragmentation in environmental stochasticity can be expected to greatly accelerate the accumulation of mildly deleterious mutations, lowering the genetic effective size. Right. So only under certain conditions does this happen. So with the study entitled Mutation Predicts 40 Million Years of Fly Wing Evolution, we explore some of these questions. Our results Here we use precise data on over 50,000 Drosophilid fly wings to demonstrate unexpectedly strong positive relationships between variation produced by mutation, standing genetic variation, and the rate of evolution over the last 40 years. Our results are inconsistent with simple constraint hypotheses. This is referring to the outdated models that the creationists are using. Inconsistent with simple constraint hypotheses because the rate of evolution is very low relative to what both mutational and standing variation could allow. In principle, the constraint hypothesis could be rescued if the vast majority of mutations were so deleterious that they cannot contribute to evolution. But this also requires the implausible assumption that deleterious mutations have the same pattern of effects as potentially advantageous ones. Our evidence for a strong relationship between mutation and divergence in a slowly evolving structure challenges the existing models of mutation and evolution. What are those existing models? Those are the outdated models that would have us believe that mutation accumulation is inevitable. So we have to add to these models processes which counterbalance mutation accumulation because we don't observe mutation accumulation of slightly deleterious mutations, at least. We observe compensation and the propagation of those genotypes with the fewest deleterious mutations. Our third result is that the phylogenetic relationship between species predicts 90% of their phenotypic similarity as measured by their phylogenetic heritability. What is this saying here? This is just a bonus result showing that the variation of the mutations that occurred to one species of Drosophila fly lined up 90% with the divergence traits of different fly species over the course of 40 million years. So this method is actually able to predict very specific predictions. This just shows just how specific and how well it correlates. This study also goes over epistasis, and we've seen it mentioned a couple times. This is essentially just the synergistic effects between two different mutations. It's related to pleiotropy, which is one mutation having two different effects, but they're slightly different. Molecular mechanisms of epistasis within and between genes. Disease-causing mutations do not cause disease in all individuals. One important reason for this is that the outcome of a mutation can depend upon other genetic variants in a genome. These epistatic interactions between mutations occur both within and between molecules, and studies in model organisms show that they are extremely prevalent. So what does this mean? It means that not only do you have a variance of alleles and mutations, but you have a variance in how those alleles and mutations interact with each other. This is just showing more definitions, specific definitions for epistasis. We're just going to deal very generally, but here are all the different descriptions for you, in case you're interested. This is again from 40 million years of flywing evolution. Or maybe it's from a different study. I think it is. It's from negative selection in humans and fruit flies and involves synergistic epistasis. So this red line here that you can see represents loss of function. All these different bars are different combinations of different combinations. If they represent the epistatic combinations interacting. So what we get is an overdispersion of variance and an underdispersion of deleterious alleles compared to what the old models predicted. So that means those models are wrong, so we're developing better models. Indeed, we detected underdispersion of the number of rare loss-of-function alleles. Thus, selection against rare protein-disrupting alleles is characterized by synergistic epistasis, which may explain how human and fly populations persist despite high genomic mutation rates. Antagonistic epistasis instead creates positive linkage disequilibrium between deleterious alleles and increases the variance of the mutation burden, leading to its overdispersion. So, overdispersion of variance in mutation burden. So, instead of just having, a variance of 1 mutation, 2 mutations, 3 mutations. No, we get this whole spectrum of how 2 or 3 mutations are interacting with each other differently according to each different mutation. So what this does is lowers the selection threshold of slightly deleterious mutations. So before we were talking about how they're invisible to selection. Well, they become visible to selection through synergistic epistasis, through interacting with other mutations. If their interaction is bad, they're going to be seen very quickly. Overlaying the mutation burden distributions with Poisson distributions having identical means shows that the underdispersion is due to a depletion of individuals with a high number of deleterious alleles. Furthermore, through regression analysis, resampling experiments, and simulations, we showed that the underdispersion signal persists after correcting for potential confounders. Okay, so that's just saying that they controlled for outliers and errors and stuff. The most neutral conformation of the nearly neutral mutant alleles is constantly selected for due to interactions between novel mutations. The epistatic model shows us that selection variance is increased due to the novel interactions between novel mutations, i.e. everyone has a unique range of mutations which behave differently with each other, with each different genome and each different mutation combination. Even individually, neutral mutations that have bad interactions with others are removed at a higher rate than if they were selected for individually. This is because epistasis lowers the threshold of selection as they build up, meaning the deleterious effects are removed before they accumulate past the threshold. The population genetics of mutations, good, bad, and indifferent. If the mutational effects are only slightly deleterious, or there are too many mutations that simultaneously segregate in a population, deterministic purifying selection can be overwhelmed by random effects, leading to a slow regime of mutation accumulation where fixations of slightly deleterious mutations occasionally happen by bad luck. If such events are repeated over long periods of time, they can have substantial consequences on fitness and may degrade the corresponding genetic system. Much research on this scenario has concentrated on cases where no recombination occurs. So again, recombination, very important for giving us new information that can compensate. It's not just removing the bad or adding in the good, it's both. Work on epistasis is important for understanding the population genetics of mutations, particularly to address questions about long-term evolution. Each species presumably lives on or near some local adaptive peak. Epistatic interactions determine how many ways there are to evolve between peaks. An increased understanding of the fitness consequences of prolonged mutation accumulation may help to predict risks to endangered species. Increases of mutation rates that may be caused by use of technology by humans may dangerously upset potentially finely balanced natural long-term equilibria between fitness increasing and fitness decreasing processes that are influenced by many evolutionary factors, including epistasis. So, while there are many factors that can tip mutations toward the good, they're finely balanced, right? So recombination and mutation rates are variable, and they're balanced to the rate of compensation. So there are some cases where too much recombination, too high a mutation rate will lead to accumulation, and there are cases where too low recombination and mutation rate will lead to accumulation of slightly deleterious mutations. Too high or too low will both result in the same result. They're finely balanced with all these different forces, such as epistasis. Calculations depend critically on the effects and rates of mutation. Yes, that's true. With increasing, if population size is small, recombination rate is low and population is fragmented. So there are all these risk factors going on here. Okay. Now, we're going to go over some specific dishonesty by creationists again. Science corrects itself. Science goes back and throws out bad models and gets new ones, unlike religion. From this study, missense meanderings in sequence space, a biophysical view of protein evolution. Quote, our understanding of protein evolution is incomplete, if not fundamentally flawed. See, we have the humility to say when we don't know something, and we don't just insert our favorite theory. The neutral theory of molecular evolution, which states that all mutations that reach fixation in a population are selectively neutral, appeals to evolutionary geneticists in part because it can account for the approximately constant rate of protein evolution. However, its premise that most missense mutations are selectively neutral has been systematically rejected by protein biochemists, who recognize instead that almost all missense mutations have large biophysical effects. Indeed, nucleotide sequence analyses have uncovered pervasive positive selection for amino acid replacements. Now, this is cited by creationists, underlined by creationists. I don't understand how. It's because they don't know what they're talking about. The premise that most missense mutations are selectively neutral has been systematically rejected by protein biochemists. So, this is the same thing. This is just a scream cap from creationists. So, if it's been systematically rejected that most missense mutations are selectively neutral, this implies that the majority of missense mutations are not selectively neutral, which means they are visible to selection. A Nephilim-free YouTube creationist was arguing with me, and I think Standing with Truth was agreeing with him, that this passage here is saying that most mutations are invisible to selection and therefore accumulate. This says the literal opposite. And later on it says here the deleterious effects of mutations are rapidly and effectively compensated by conditionally beneficial mutations. Biochemical reality that missense mutations have large context-dependent effects that few, if any, are selectively neutral. This goes against the creationist claims. By the way, the most missense mutations are visible to selection, and that proportion seems to be about 70%. The average proportion of deleterious amino acid polymorphisms in samples is about 70%. That's from prevalence of positive selection among nearly neutral amino acid replacements in Drosophila. So, this is the mind of a pseudoscientist. You read that most missense mutations are not selectively neutral, and from that information you somehow get that most missense mutations are selectively invisible, aka neutral. You're literally dealing with someone who thinks up is down. But it's all about recombination. That's recombination promotes genomic integrity among cells and tissues through double-strand break repair and is critical for gamete formation and fertility through strict regulation of the molecular mechanisms associated with proper chromosomal disjunction. Recombination is among the most prominent mechanisms shaping genome variation and is associated with not only the structuring of genomic variability, but is also tightly linked with the purging of deleterious mutations from populations. That's from the impact of recombination on human mutation load and disease. I'm not just talking out of my ass, these are scholarly sources. But I'm also talking from my own experience. These are all arguments built from my education. I just went and got sources to confirm them all. So we've been talking about compensatory mutation, the rate of compensatory evolution. This paper states, in proceeding under mutation pressure, natural selection, and genetic drift from one fitness peak to another, a population must therefore pass through a valley of intermediate deleterious states of individual fitness. The expected time for this transition is calculated using diffusion theory. The rate of compensatory evolution, Kc, is then defined as the inverse of the expected transition time. Now the rest of this goes over some more specific math. The creationists are welcome to challenge this math. It's fairly simple as far as math goes. So is this math. This is also describing the math behind this. These are about the ratios and balances of the different forces. So we can assign mathematical behaviors to rates, for example, rates of mutation. We can assign values to the variance. We can assign values to any of these modifications and selection pressures. And we do. And creationists don't challenge these models at all. They just argue in rhetoric. So, compensatory mutations are pretty interesting. Because they are nonsense, but they occur within this range of stability. So due to anti-fragility, you can change things slightly and they'll still retain most of their original function. You can mess with them. They go up and down, it's still stable, still has mostly the same function. Some mutations will simply bring a bad mutation into this stable zone. Some mutations will just oscillate a single gene within the stable zone. Some mutations will take this stable protein and make it deleterious, or make it lose its function. Other mutations I've shown, and we're going to go into very specific examples, can restore the stability and original function, or, this would be a new function. The near neutrality in evolution of genes and gene regulation. In particular, the evolution of regulatory elements of duplicate genes is likely to be nearly neutral. It's important for preserving duplicate gene copies and for the evolution of new functions. Positively selected mutations include adaptive mutations, i.e. mutations that directly affect enzymatic function, and compensatory mutations, which suppresses the pleiotropic effects of adaptive mutations. Compensatory mutations are by far the most frequent of the two and would allow potentially adaptive but deleterious mutations to persist long enough in the population to be positively selected during episodes of adaptation. So you can actually get a few deleterious mutations accumulated alongside beneficial mutations, and they can actually be selected for independently. Later on, because if a organism with one beneficial mutation and 3 deleterious mutations breeds, it's going to produce a variance of organisms. It's going to pass on its beneficial mutation to most and its deleterious mutations on to most. But there is going to be one of those combinations that has only the beneficial mutation and none of the deleterious mutations. And this process can repeat. Not only that, but compensatory, but you can back mutate, and you can mutate compensations, and these occur frequently. Compensation occurs more frequently than back mutation. So it seems as if life has a preference for progression and change. The nuclearly recognized relevance of thermodynamic stability for function has obvious implications for protein modeling and engineering. It has dramatically improved our ability to identify critical functional residues in a protein sequence and to predict the functional impact of mutations. Enzymatic stability considerations should become key for protein engineering. Consensus design based on phylogenetic comparison has already been used to improve enzymatic stability. So there you go. Again, understanding of evolution is helping us actually produce technology in real time. Genetic constraints on protein evolution. Adaptive mutations frequently have deleterious pleiotropic effects, and these are suppressed by compensatory mutations. This is the literature. Creationists ignore this literature. They don't even want to talk about compensatory mutations. Mutations that are pathogenic in one species are often fixed in another, suggesting that the deleterious effects of missense mutations have been compensated by other mutations. That, or it's suggesting that it has different synergistic epistatic effects with different genotypes. So again, protein function is entirely context dependent. It's not like you have a broken gene versus a working gene. This isn't a broken script versus a working script. They have different functions. If I wanted to use the resetting countdown flag for the pausing countdown flag game, yeah, then it would be broken. But it's context dependent. See, I have two different game types, and each flag has a place in each different type. A recent convergence of directed evolution, population genetics, genomics analysis, and experimental adaptation experiments have challenged traditional notions about protein evolution. Positive selection has been recognized as an important driver of evolution. A large portion of positively selected mutations appear to be compensatory, suppressing deleterious pleiotropic effects of adaptive mutations. Neutral mutations are neutral because they have little effect on function. So, there are missense mutations that occur. Most of them cause, 70% of them cause bad effects. 10% of them do not, because those mutations are occurring in genes that don't have a huge role, or genes or those mutations do not have a huge effect on the role of that gene. And so these are easily compensated for. Deleterious effects of adaptive mutations are subsequently suppressed through the fixation of compensatory mutations. Typically, suppression occurs within the time frame of adaptation and often involves multiple compensatory mutations. These compensatory mutations facilitate adaptation to novel environments by precluding reversion to wild type once selective pressures are removed, and by prolonging the lifespan of adaptive mutations in the population. So we wonder why things don't evolve back the direction they came from, even though it works. It's because of compensation. Because you can't go back on compensation. Because then you would be reducing fitness, and you would never be selected for that way. You can create children with less fitness, but that's not going to progress the population in that direction unless it compensates. And so the fact that compensatory mutations will stop backsliding because it adds to the complexity of the backslide. You have to delete that compensatory mutation and get a back mutation. And that's less likely than just having another slightly deleterious mutation, which occurs 10% of the time, have a compensatory synergy. Now, compensatory mutations are important in a very typical creationist misunderstanding of evolution. So we actually see wild type pathogens, viruses and bacteria, evolve in their wild environment, aka our bodies, basically, and hospitals. We see this in the lab too. We see them evolve antibiotic resistance. And creationists understand and they accept how this happens is random variation will create a mutant that has a protein that will confer resistance. And they point out, half correctly, that these mutants take a hit in fitness. Now, this is observed in some laboratory settings. It is not observed in the wild. So, but also in the laboratory settings, what we observe is compensatory mutations suppressing the hit in fitness. Compensatory mutations, antibiotic resistance, and the population genetics of adaptive evolution in bacteria. In the absence of selecting drugs, chromosomal mutations for resistance to antibiotics and other chemotherapeutic agents commonly engender a cost in the fitness of microorganisms. So, yeah, we do observe the evolution of microbiotic resistance or antibiotic resistance can engender a cost in fitness in microorganisms. Sure, we see that in the lab. Recent in vivo and in vitro experimental studies of the adaptation to these costs of resistance in E. coli, HIV, and Salmonella found that evolution in the absence of these drugs commonly results in the ascent of mutations that ameliorate these costs, rather than higher fitness, drug-sensitive revertants. The rate of compensatory mutation being substantially higher than that of reversion. So, what's happening here is we're observing compensatory mutations ameliorating the cost to fitness that the original antibiotic resistance gave us. So, the hit to fitness is only temporary. Just requires more evolution. And I've explained that to the creationists before. Here is an actual study observing it happening, so you have no wiggle room here. None. The X chromosome and the rate of deleterious mutations in humans. Basically, this is just estimating the accumulation of 4 deleterious mutations per human zygote per generation, and suggests that the continued existence of our species probably depends on quasi-truncating selection and recombination. This is pretty low buildup, especially given the rate of compensatory mutation. And what is the rate of compensatory mutation? Well, there's a study done on the DNA bacteriophage X174. For deleterious mutation, we evolved 8 to 16 replicate populations to determine the frequency at which compensatory mutation instead of back mutation was acquired to recover fitness. There's that type of experiment again, recovering fitness from another adaptation, such as antibiotic resistance. The overall frequency of compensatory mutation was approximately 70%. So 70% of the time there was a compensatory mutation. So going back here, it doesn't matter if we get 4 deleterious mutations per generation, because 70% of us are going to compensate for that. At least they're going to develop a type of compensatory mutation. It's not clear whether this is a successful compensatory mutation or just another neutral. I think what it means, if it's a compensatory mutation, thus it did compensate. It was shown to compensate. I'm willing to state that is the correct interpretation. The overall frequency of compensatory mutation was approximately 70%. Deleterious mutations that were more severe and were significantly more likely to be compensated for. This is observed in real time. Moreover, a likelihood analysis of mutation diversity predicts that, on average, a deleterious mutation can be compensated by about 9 different intragenic compensatory mutations. So, where were we? Okay. So, not only is the rate of compensatory mutation very high, the rate of slightly deleterious mutations very low, the rate of removal of very deleterious mutations and compensation for very deleterious mutations is much higher. Not only that, but for every deleterious mutation, there were about 9 different ways to compensate for it. Here's more examples of compensatory mutations. Restoring allosterism with compensatory mutations in hemoglobin. Allosterism is basically the ability of a protein to change conformation due to one or multiple substrates. So it will actually have a different function depending on what's attached to it. So we lost allosterism in hemoglobin due to a mutation, and then due to a different mutation, we restored it. This paper states, our results clearly show that the tyrosine alpha 42 aspartate, the tyrosine alpha 42 2-aspartate replacement can substantially compensate for the functional defect of hemoglobin chemistry caused by the aspartate beta 99 asparagine 2-asparagine substitution. The structural and functional information derived from this recombinant hemoglobin provides insights into the structural basis of allosterism and the design of compensatory amino acid substitutions to restore the functional properties of other abnormal hemoglobins associated with hematoglobinopathies. Hemoglobinopathies. So, structural and thermodynamic analysis of compensating mutations within the core of chicken egg white lysosome. The results illustrate how proteins can compensate for potentially destabilizing substitutions in different ways and underscore the importance of high-resolution structural data. The findings also suggest that protein stability can be increased by mutations that lower the strain in the protein interior while maintaining total buried hydrophobic surface area. So, this is proteins, chemicals evolving, but it is evolving due to life replicating. So don't confuse this with chemical evolution. Never do that. I'm actually kind of sorry I brought it up. Compensated deleterious mutations in insect genomes. These results support a selection-driven process that allows compensated amino acid substitutions to become rapidly fixed in taxa with large populations. So we're observing this everywhere. This is a duplication, right? I think that's a duplication. Okay, the X chromosome and the rate of deleterious mutations in humans is a duplication. Compensatory adaptation to the deleterious effect of antibiotic resistance in Salmonella. Most chromosomal mutations that cause antibiotic resistance impose fitness costs on the bacteria. This biological cost can often be reduced by compensatory mutations. In Salmonella typhimurium, the nucleotide substitution AAA42 to AAC in the RPSL gene confers resistance to streptomycin. The resulting amino acid substitution in ribosomal protein S12 causes an increased rate of ribosomal proofreading, and as a result, the rate of protein synthesis, bacterial growth, and virulence is decreased. 81 independent lineages of low-fitness K42N mutant were evolved in the absence of antibiotic to ameliorate the costs. Only 4 of the evolved lineages contain streptomycin-sensitive revertants. The remaining 77 lineages contain mutants that were still fully streptomycin resistant, had retained the original resistance mutation, and also required compensatory mutations. Our results show that the deleterious effects of a resistance mutation can be compensated by an unexpected variety of mutations. So, just study after study of observing this in real time. Compensatory mutations, antibiotic resistance, and the population genetics of adaptive evolution in bacteria. The results of our investigation support the hypothesis that in these experiments the ascent of intermediate fitness compensatory mutants rather than high fitness revertants can be attributed to higher rates of compensatory mutations relative to that of reversion and to the numerical bottlenecks associated with serial passage. So, the higher rates of compensatory mutations are probably due to the fact that a back mutation or reversion has one chance to happen in the exact same place that we just had a previous mutation. Compensatory mutations can occur in at least 9 different zones. So 9 possibilities to 1 possibility. Of course, you're going to get more compensatory mutations. Now, we're starting to wind down here. The genetic costs of Neanderthal introgression. In this paper, I believe it states that Neanderthals were 40% less fit, and creationists will confirm this. They say they parrot this a lot, especially to suggest that Neanderthals are degrading faster than humans, although that somehow betrays the fact that we're able to stop that degradation somehow. But I'm more interested in the fact that they can detect Neanderthal DNA at all. Also in this paper, increased load predicts the reduction in Neanderthal introgression around genes, these into human genes. So this does not mean that the the genome degradation is predicted. It means that it predicts Neanderthal gene transfer into humans is slowed down. That's what introgression is. Reduction in introgression is slowed down. This supports the opposite of inevitable genome degradation, since the genes are less fit, and we see a slowed down introgression into the human genome. This is proof that the genome is able to slow down and reduce harmful mutations from entering the gene pool. Most young Earth creationists have already admitted Neanderthals are humankind, but their predictions should show that they branched off from some Adam-like perfect human. Instead, we only find ape-like hominids prior to Neanderthal. Plus, this admits the concept of comparative genomics. If you're admitting that we can detect Neanderthal DNA in human DNA without ever seeing a living Neanderthal, let alone seeing a Neanderthal breed with a human, then you're admitting that we can detect ape DNA in humans. Do you get it? Here's another real-world example of evolution acting extremely quickly. These butterflies were infected by, I think they were the Wolbachia bacteria, and they were killing off males in their larval stages. So there were like 90% females or something, And there was one butterfly that developed a mutation that gave it a resistance to this bacteria in its development, and within a couple generations, the male and female populations were back to normal. This gene had spread throughout the entire population, but had not happened on a population on a different island. Pleiotropy, we went over a couple times, is essentially just it's similar to epistasis, but it's specifically where one gene affects two or more traits. And this is all building up to anti-fragility. The fact that we can mess with the system and still get something. The genome seems so intricate and delicate, but the systems are actually less fragile than you might think. We can get a two-headed animal that can live its whole life. Now, this isn't an example of a very beneficial mutation, but the fact that you can mess with the system so much and yet it won't crash is proof of its flexibility. Even the basic components of RNA synthesis and translocation, metabolism, even these basic proteins are different between two different species of bacteria. Showing that you can mess with the fundamental synthesis molecules of DNA. There's variation among the fundamental cellular, transport, and transcriptional proteins. The notion of anti-fragility, an attribute of systems that make them thrive under variable conditions. This idea requires the ability of such systems to tinker, i.e. to creatively respond to changes to their environment. One such example is proteins with flexible regions that can undergo functional alteration to their side chains, side residues, or backbone, and thus implement the tinkering that leads to anti-fragility. We've seen this multiple times. So you can change something slightly and add a function or change its function. Now it might have two functions. Might be pleiotropic now. And of course, it's always context dependent. The same information is flexible enough to do two different things in two different contexts, and one context may be more functional than the other. As I mentioned earlier, I make games and weird stuff happens. They're called glitches. They sometimes crash the whole system, they sometimes just break and do nothing, but the rest of the game just functions just fine. Sometimes they give really funny effects, sometimes you can barely notice the glitch, sometimes the glitch actually gives us a function we want, or a constraint that works, or an effect that was totally unexpected but welcome. For example, we had these seats that you could attach to a chassis, and they had their height parameters removed, so you could only run vehicles on them on a flat plane. I was trying to make carriages and cars from this, and I considered it a broken script. But this allowed us to make floating airships or moving platforms, which would have been totally broken if the height adjustment was there. So, broken for cars, not broken for airships. But the seat that would have made cars move would have been broken for airships. Or moving platforms. Or moving bridges, kind of. Young Earth creationists would say that's a broken script. No. It can be repurposed in a different environment. It is context dependent. What's the likelihood of an individual mutating to have a suppressor function specifically to target the genetic dysfunction inherited in the population? Unlikely, individually. But if, by bad luck, a slightly deleterious mutation becomes fixated, there are millions of opportunities for a compensatory mutation to develop, and this will rapidly fixate. And like we said, there are about 9 chances per deleterious mutation. Compensatory mutations occur at a rate of 70% for some functions, for each individual, I think. So for each function, the likelihood of a compensation in a healthy population is very, very high. We're talking about the population, right? So we already have the mutation fixated in the population. Now, all the individuals have this 70% chance to produce a compensation. Now, young Earth creationists will claim that since it takes so long for one beneficial mutation to correct a bad one, while deleterious ones accumulate, again, they ignore that they are nearly neutral and that most neutral conformation is always selected for and will be compensated for before it can accumulate. Or it will be selected out before it can accumulate, as with epistasis. Most mutations are serious and visible to selection, about 70%, and slightly deleterious mutations represent about 10% of the total, with compensatory mutations occurring at a rate of about 70%. So not only are compensatory mutations more likely to become fixated, but they occur at a higher rate than slightly deleterious mutations do. Every time a mechanism is presented that refutes their claim that something cannot happen, young Earth creationists move their goalposts back. Anti-fragility, epistasis, and all the different kinds. Genetic purging, anti-fragility, modularity, genetic drift, balancing selection, directional selection, disruptive selection, fluctuating selection, stabilizing selection, sexual selection, coinophilia, dysgenics, mutation selection, balance, and compensatory mutations. All of these are ignored and thrown out the window. Simply said, well, you can't get rid of slightly deleterious mutations because they're selectively neutral. Even though we just showed you several processes by which they become selectively non-neutral, selectively visible, or compensated for. Review of compensatory mutations. There are more papers on compensatory mutations because we can observe them and are happening right now in real time, and they appear to have a huge effect on stymieing Miller's ratchet. Epistasis reduces the deleterious mutations that become fixed, and compensatory mutations erase the effects, suppress the effects of what sneaks through. Or in other words, recontextualizes the effects of what sneaks through. None of the creationist literature is cited by anyone but other creationists because no one finds it useful, because the only thing that creationist literature is good for is spreading creationism. It has literally zero applications outside of making it look like creationists are able to challenge science. But once you get to God did it, there's nothing left to talk about or to discover. The reality is there's plenty to discover, and God did it is just putting your mind in a prison and blinding yourself to new information. I guess we'll go over the greatest hits here. Well, I've already read all of them, but these are for you, Neff. But these are common misinterpretations of evolution. So I was challenged to say that the majority of accrued deleterious mutations in any given species have been observed to be removed by the populations. I was challenged to show me you will never in your life find one statement like that in any science paper. I believe I'm about to read off about 11 for you, Neff. So, recombination is among the most prominent mechanisms shaping genome variation and is associated not only with the structuring of genomic variability, but is also tightly linked with the purging of deleterious mutations from populations. Negative or purifying selection prevents the unlimited accumulation of deleterious mutations and establishes a mutation selection equilibrium. Thus, average human should carry at least 7 de novo deleterious mutations. If natural selection acts on each mutation independently, then resulting mutation load and loss in average fitness is inconsistent with the existence of a human population. Overlaying the mutation burden distributions with Poisson distributions having identical means shows that the under dispersion is due to depletion of individuals with a high number of deleterious alleles. Indeed, we detected underdispersion of the number of rare loss-of-function alleles in 8 independent datasets from human and fly populations. Thus, selection against rare protein-disrupting alleles is characterized by synergistic epistasis, which may explain how human and fly populations persist despite high genomic mutation rates. The overall frequency of compensatory mutation was approximately 70%, while only about 10% or fewer of mutations seem to behave as slightly deleterious mutations. A deleterious mutation can be compensated by about 9 different intragenic compensatory mutations. Deleterious mutations that were more severe were more significantly significantly more likely to be compensated for. Our results clearly show that tyrosine alpha 42 to aspartate replacement can substantially compensate for the functional defect of hemoglobin chem-C caused by the aspartate beta 99 to asparagine substitution. That's from Restoring Allosterism with Compensatory Mutations in Hemoglobin. So, what's with this red herring? What do we keep circling around? The claim, the accumulation of slightly deleterious mutations cause unavoidable genetic decay and dysfunction without exception, effectively putting an imminent expiration date on all life, a process which is about 4,000 to 6,000 years old. Evidence for this? Nothing. Absolutely diddly squat. Even by the research that they cherry pick and cite, we would have died 100 times over. We would have died, well, at least under normal timescales, but even on the creationist timescale, we would have died 3,000 years ago. Or 1,000 years ago at the youngest. Everything I just explained, young Earth creationists will ignore because it counters the directionality of Muller's ratchet and makes it less likely to occur and to be outweighed. Back mutations occur, reverting to wild type, but compensatory mutations occur at a higher rate. Most mutations are not silent to selection and are removed from the population quickly. Most silent mutations are removed quickly, too, because of if they're slightly deleterious, because of epistasis. Compensatory mutations are shown to occur at rates of 70% for slightly deleterious mutations, compensatory and at a higher rate for more deleterious mutations. Compensatory mutations are shown in numerous observations to suppress deleterious ones. Pleiotropy shows us that mutations affect each other, so does epistasis, and thus have different effects next to other mutations. Epistasis lowers the selection threshold of accumulation and increases variance of the populations. These are just some of the very powerful forces that are known to counteract the direction of genetic entropy, or the genetic load paradox. Genetic decay paradox, I'm sorry. Young Earth creationists will see that individually, none of these is powerful enough to turn the tide, but in typical fashion, they are in denial that these things can add up. And this time isn't even evoked. These are all stacking at once, and these are all observed in real time. Problems with the biblical model. Mutations introduced after the first life forms, meaning we lived without mutation before, meaning it's a magical curse and not the regular interaction of chemistry. As we understand it, mutations occur due to the actual kinetics of the molecules. So if they're not in the right position, they won't bond right, and thus errors occur. That's what mutations occur from. So creationists either have to explain how God was stopping that from happening prior to the fall, or how God induced that to happen after the fall. In the biblical model, we would not see living fossils. Stasis would be impossible without proving that evolution can last over thousands of years, because if stasis is possible, then we can just stay in stasis. And if stasis is possible, that means there are forces that are delaying and halting Muller's ratchet, the accumulation of slightly deleterious mutations. Young Earth creationists admit that mechanisms can slow and speed up mutation accumulation. But all they'll say is, oh, it just slows it down. Slows it down ever so slightly to the fact that we see no change in 6,000 years. Even though these relationships have accurate mathematical models, young Earth creationists reject the conditions which keep weakly deleterious mutations from accumulating. They refuse to investigating because using big science words to confuse people works better than actually learning math and actually learning biology. Created heterozygosity and differentiation of traits from that point is still evolution. Concluding. Accumulation. In all these cases, mutation accumulation alone is not sufficient to lead a population toward any kind of foreseeable extinction event, just like beneficial mutation alone is not enough to lead the population in the opposite direction. Unstable environment, mutagens, and small populations are explained as the biggest variables that can tip the balance toward extinction and towards Muller's ratchet. Young Earth creationists will not challenge the math behind this. They will simply deny that these conclusions are scientifically based. Beneficial mutations outweigh deleterious ones in certain conditions that we can model. You have to show how these models are unrealistic, but you can't because these models make the predictions that we see and that are confirmed in the wild and in the lab. By the way, none of this even matters because creationists already admit all the mechanisms of basic evolution, and they admit all the mechanisms for slowing the accumulation of deleterious alleles. On top of all of that, we observe persistent populations, we observe populations millions of years old, and we observe populations in stasis, and we observe mutations being removed and compensated for. And on top of that, they admit comparative anatomy and comparative genomics. So we don't even need any genetics. We don't even need dating in the fossil record. But we have it. The question becomes, so what is the extent of the change among life? What's the extent of evolution? They've already admitted evolution. So what's the extent of it? What was the first life? If only there was good evidence or even a recording of what actually happened so we can observe the evidence ourselves without speculating. If only there was a camera that took a picture of the populations as they changed over time. Oh, there is. It's called the fossil record. Well, anyway, differentiation is evolution. It is changing population traits over time, and this is linked to genes, and creationists admit this. Even they just conflict with the mainstream evolution on their assertion of an absolute constraint on evolution that magically prevents kinds from evolving into new kinds, and that deleterious mutations accumulate without exception. All scholarly sources debunk these unobserved conclusions from outdated and misinterpreted models. All they want to tell me, and they want to tell me that hip bones getting a little smaller, or the nose moving up the snout, or hands getting a little stiffer, as in the case of whales evolution, is an evolutionary leap of diversification that's too far, but the bone structure of all these different dogs changing drastically is not an evolutionary leap. Young Earth creationists accept paternity tests, but not common ancestry. They even accept paternity tests between Neanderthals and Homo sapiens. But they can't accept it between Homo erectus and Homo sapiens. They can't accept comparative anatomy between them either, even though they accept comparative anatomy between cats. This is what it's like to deal with the mind of a delusional pseudoscientific ideology. So once you've shown them all the sciences, they have one thing to do, and that is to move the goalpost once more and say, well, you never actually saw any of these changes occur, so even though you can show that they happen in modern animals, you can't show that they happened between fossil animals. So that's what we're going to be covering next time.